Hello, everyone. Thank you for joining this Mission Bio webinar focused on software and bioinformatics for single-cell DNA analysis. My name is Anup Parikh. I lead the product management group here, and I have the honor and pleasure of talking to you about all of the work that we've been doing here around software development and bioinformatics methods development to really getting insights into single-cell biology. Today, I'd like to do three things with you. One is to go through why we're doing what we're doing. Second is to give you a little bit of a flavor of what our customers are doing today with the Mission Bio platform in cancer research. And then lastly, really focus in on the software that we have available to all of you to be able to do this research yourself. So let me get started here. Just to put this into context for us, we really focus on cancer research, and 1 in 3 of us will be affected by cancer sometime in our life point. This is a really personal disease. This is a really global problem that we have to deal with, and it's a big challenge that we have to deal with. Cancer isn't a single disease. It can happen in any of our 37 trillion cells. It can happen in many different organs. So this is a large problem. But the scary part of cancer is that it only takes a single cell with the right set of mutations to start the disease. Being able to identify this single cell and see it progress into the disease that's ultimately going to affect 1 in 3 of us is a really big challenge that we want to help tackle. Now, looking at cells isn't a novel thing. Cancer is a genetic disease, but it causes dysfunction of cells. And in medicine, we've been looking at cells for hundreds of years now. We've been looking at slides like this where we can stain cells and look and see that the purple cell here is abnormal versus the rest of the cells. And this is how we've been dealing with detecting and treating and monitoring cancer for decades or centuries now. In the last 30 years or so, genetics has really increased our ability to go deep and understand the basis of this disease. But we lost the context of the cell when we started doing genetics. We combined all the cells together, we lysed them, and we looked really deep into the DNA of these cells or the RNA of these cells, but we didn't retain the context of the cell. With the revolution that we're having in single-cell genomics these days, we can start marrying these two concepts together now. We can retain the concept of the cell or the context of the cell, which is what we've been used to for centuries now, but also now look deep into each one of these cells to detect what is happening at the genomic level, the transcriptomic level, or the proteome. This is a really powerful set of data that can help us elucidate what is going on in the cancer in a way that we've never been able to do before. Now, specifically at Mission Bio, we're really wanting to focus on unraveling the complexity of cancer, looking at the DNA. This DNA at the single cell level really helps you measure tumor heterogeneity. It's looking at the fundamental building blocks of each one of these cells. This is where all of the pathogenic mutations that we're used to, the biomarkers that we're used to for cancer research, whether it's in diagnosis or in treatment or in targeted therapies or in monitoring, we're used to looking at the DNA. And we want to look at this heterogeneity across thousands of single cells. We can do this at single cell level by tracking the evolution of these subclones and be able to detect really rare subclones down to 1,000. Where our focus is looking at the DNA and unraveling the complexities of cancer. Before I go into the details of what we do, we had the pleasure of having Dr. Elliot Stieglitz in our offices a couple of months ago talking about this rare disease. And I wanted to just share this disease to, again, personalize what is going on and why we do the research that we do. So juvenile leukemia here, it's a rare form of leukemia that affects young children. It's a really good model organism to study leukemia, and that's why it's been used quite often to study the basis of disease formation. And really, when Dr. Stieglitz was in our offices, the challenge that he put in front of us and presented on was could we tell the difference between these two children that were enrolled in a clinical trial, both of them with this form of rare leukemia? And what we do is really to try to understand why these two children might have the disease. How can we distinguish one that might be able to survive with a certain therapy while another one might not be able to? And when we just look at standard pathology without genomics, the survival curve here is about 50%. So one of these two children is going to survive. We can't really tell which one of them. And we really need to develop better resolution technologies, which is where genomics has been fantastic over the last 30 years. Once we add genomics and look at the canonical mutations across these patients, we can start seeing the group segregate by the type of mutation that they have. This is great and gave us a lot of information. But again, back to these two children, we can't tell the difference because they both have the same canonical mutation, and we can't predict which one of these two will survive just on that one mutation. When we layered on the context of the cell, looking at whether there was one or two alterations in a cell or more than two alterations in a cell, you start seeing a nice segregation between survival and relapse. And now, when you look at these two children, you can see that the child on the right has a double mutation, that two mutations occurred in one of the subclones, which unfortunately made that child unresponsive or relapsed to the therapies that were being tested in that clinical trial, whereas the child on the left survived with that single mutation. So that's really the power of single cell context. It allows us to get better resolution to be able to answer this question of who's going to be affected by the disease, who's going to respond to treatment, and who might eventually relapse. So our mission statement as a company is to really empower researchers and clinicians to unlock this power of single cell biology to enable discovery, development, and delivery of precision medicine. And we're doing this by developing very targeted solutions that address high-impact applications like cancer research. So as a company, what we do is we develop instrumentation, we develop targeted assays, and full end-to-end software to be able to interrogate genomics at a single cell level. Our workflow spans all the way from designing targeted assays, specifically targeting a set of mutations or a set of genes that are relevant to a cancer, being able to do library prep in this microfluidics device that allows us to encapsulate individual cells and tag them with a cell barcode so that we can retain the single cell resolution. Sequencing is done on standard Illumina sequencers, and then we have a full suite of analysis solutions to be able to interrogate and get insights from the data. Just to give you a flavor of what kind of data is ultimately generated from our solution, this is a fish plot showing a sample which was from pre-treatment, on treatment, time point 1 and 2, and then relapse. So this is a serial time point where we're looking at a set of specific subclones that were discovered in this patient. In this case, there were 3 different subclones that were relevant. Each one of these subclones is essentially a group of mutations that are found in a set of cells. So the blue subclone is one that initially caused the disease. There was a treatment that was given that caused that subclone to decrease in population, but ultimately there were 2 other subclones that increased in population because they were resistant to the original treatment that was given, which caused the relapse. This is actually a fairly simple counting problem. We just look at the set of cells that we've pushed through the Tapestry system, identify which mutations occur in each of the cells, and then count the number of cells that have that mutation profile. So, and then put that over a longitudinal view in this fish plot. But this gives you a really powerful view of what's going on over time, how the subclones now are evolving over time due to various treatments or interventions. Now, this paper has just recently been published in Cancer Discovery, where they looked at multiple different patients across the serial time points. And you can see in some cases, in the case 1, a subclone that ultimately relapsed was actually detectable pre-treatment at a very low number of cells. So the resolution that we can gain by looking at 1 in 1,000 cells allows us to look at these cells very early on and say, hey, this is something that existed pre-treatment, ultimately leading to relapse. You can look at it early in treatment because, again, the resolution is really high. And then the last example, this is just showing kind of the combination of NRAS mutations that ultimately led to relapse. So it gives you a power to really look at evolution that might be happening in many different ways, whether it's early in the disease or later in the disease. Another example of this type of work from another one of our customers or early collaborators, looking at specifically post-treatment, whether we have relapse happening during the remission stage. So we're looking for signatures of minimal residual disease. In this case, the first example is a fairly simple example where you have a disease in the red slice, and ultimately that same subclone relapses because there were a handful of cells that still survived during the remission stage. The middle graph really is looking at a very complicated evolutionary path where you have many subclones during the diagnosis stage, and then also many subclones that still are retained during the relapse. The third case is a really unique case in the sense that all of the subclones that were there during diagnosis were treated for, but then a brand new subclone with a different set of mutations, the green subclone, was ultimately the cause of the relapse. So this is another flavor of how the technology and the analysis solutions are used to really look at the evolution of the tumors or the cancer over time and how these subclones evolve. Now, both of these examples were from liquid tumors, from leukemias. We've also done this in solid tumors. So this is an example of work where we isolated nuclei from fresh frozen tissue and looked at different samples from a patient where different organs were biopsied. And here you can see an evolutionary tree or a phylogeny across the different tissues, and you can see two distinct branches where from the origin of the cancer you see two different evolutionary paths or two different sets of mutations that give rise to different tumor in the wall of the heart versus the liver. And you can also see in this right panel that in the normal liver tissue, there's actually a very small number of cancer cells that are still present and detectable by the Tapestry system. Again, showing the power and the resolution of being able to look at physical cells and detect 2 or 3 or 4 cells out of a population of 1,000. Another example of where Tapestry's been used is in gene editing. This was an example of an early collaboration where a customer, an industrial partner, came to us and said, we're trying to make edits within a cell line. In this case, this is a T cell line. And they were trying to make 3 specific edits where doing just bulk sequencing, as you see in the left panel, you'd be able to see the efficiency of those 3 edits as it's shown in the bar chart. But when you look in the middle panel with the single cell resolution, you can see not only the edits, but the combination of edits that took place within each cell, and then whether it was homozygous or a heterozygous edit in each one of those cases. So there's a total of 27 different combination of edit possibilities with just 3 edits that you're trying to make, and we can try to resolve all 27 of those with single cell resolution. So this gives a lot more insight into, are the edits being made the way we expect them to be made, and ultimately optimize the editing system to give us the right optimization that we need. So this is just a flavor of a few of the ways that we do, the way that people use the Tapestry system to be able to interrogate in heme cancer, solid tumor cancers, as well as in the gene editing space. And really what I want to do in the next set of slides is walk you through how we've developed analysis solutions or software solutions across the whole continuum of designing targeted assays all the way down to how do we interpret the data. Let me move to the next slide. So across our entire workflow, we've developed 3 sets of softwares that are available to you today. The Tapestry Designer software is a web portal that allows you to design, that enables you to design custom DNA panels to interrogate specific genes that you're interested in for your disease research. The pipeline, Tapestry Pipeline, is again a web-based portal that is enabled to cloud computing that allows us to process the FASTQ files from the Illumina sequencing for the thousands of cells that you're processing on the Tapestry instrument. And then the Tapestry Insights tool is a desktop tool that takes the variant calling data and allows you to filter, annotate, and visualize that data so that you can ultimately gain insights and interpret the results that came off of the individual samples. And I want to walk through each one of these in detail, give you a little bit of a flavor of how we've developed the software, as well as go into the details of the bioinformatics pipelines that are on the back end that enable you to do this complex work with a very simple and intuitive user interface. So starting with the Tapestry Designer, this is our front-end portal to be able to design specific targeted panels. We have a set of these panels that are already available on this tool for different heme cancers, as well as universal solid tumor panel. And then the tool also allows you to bring your own custom or your own set of targets and design the set of amplicons for your own assay on the fly there. So both of those features are available on the Designer tool and available today. So when you log into the tool, you'll have the ability to choose whether you want a panel that we've already designed or whether to start your own custom panel. When you look at each one of these panels that's been designed, we spent a lot of time on the back end developing the specific algorithms that will generate the right set of amplicons that will work together in this multiplexed PCR and give you as uniform an answer as possible. And all of that bioinformatics expertise is bundled into this nice intuitive front end where when you look at the panel, you're just seeing the series of amplicons that have been designed, exactly where in the genome they've been designed to, and if there were regions that you had asked for that we couldn't cover, then we'll also clearly define why those weren't covered or where they weren't covered. So in this case, this example that you're looking at, about 2% of the targets that were requested weren't covered, and we clearly state that in the tool itself. There's a downloadable zip file with all of the data available if you're bioinformatically inclined to be able to go and look at that on your own. We also have a link directly to the UC Santa Cruz web browser, so you can look at all of these specific amplicons that we've designed in the context of all of the other annotation that the browser, UC Santa Cruz browser, enables. So you can really judge whether the panel that we've been able to design meets the criteria for your experiment and whether there are any changes that you want to make. And once you've done that, we've got the ability to order right from the website, so that somebody from our team can contact you and start the manufacturing process. Now, we hide a lot of the complexity behind what we're doing on the back end in this simple interface, but I want to give you a little bit of a flavor of what we do on the back end. The challenge of designing a custom assay is both in the in silico design of the amplicons that you want to target the specific regions, and then also in the manufacturing of those amplicons so that they behave well together to give you as uniform a panel as possible. And so we want to tackle both of those challenges together with this simple interface. And I'll just give you one example of how we're using all of the data that we've generated to date to improve on the quality of both the in silico design as well as our manufacturing capabilities so that we can, on the first pass, deliver the highest quality panel back to you, even though it's a custom panel just specifically to your targets of interest. So in this data slide here, we're looking at, I think, a panel of about 300 amplicons. And everywhere you see a yellow dot is one where the amplicon was not performing at the quality that we expect to be able to call variants from that amplicon. And so the initial panel that we had developed had about 70% high quality amplicons, 30% of them were low quality, highlighted in yellow. We applied a simple machine learning algorithm to this large data set, or fairly large data set, about 300K points, to identify features that are specifically related to quality. And using those features, go back and re-optimize which amplicons are likely to work best in this multiplexed PCR. And using that, we were able to improve the quality of the amplicons from 70% to 90%. The features that we found are not that novel. These are things that any of us who have done multiplex PCR have seen before. GC content makes a big difference in the quality of the panel. The complexity of the DNA there, kind of the mappability or the repeat elements, these are all things that cause the quality of the panel or the quality of the amplicons to suffer. And we try to build all of this into our bioinformatics pipeline on the back end of the designer tool so that it's doing its job, it's kind of transparently to you to be able to design the set of amplicons that we think are going to work as best as possible. Now, a future task for us is to use even more of this data as we're generating more and more custom panels to improve even further this 90%, with our goal is to get to 98% of all of our amplicons working as well as expected. And we're going to do this in a couple of ways. One is to utilize all of this big data that we're developing or we're generating to improve on the algorithm so that we're not just using basic machine learning algorithms, but we can really start using some of the deep neural network algorithms that are out there that can make use of the big data in a much better way. And then what we also would like to do is to be able to give you a prediction up front of what this algorithm thinks it can generate so that you can make the decision about whether a certain amplicon or set of targets is important to you and you're willing to accept a lower performance rate versus other amplicons. So it gives you control and flexibility in deciding quality versus the content that is really critical for your research. All of this, again, will be hidden behind the simple user interface that's available to you on the designer tool today. The second tool that we have, once you've built your assay and run your samples through the Tapestry system to generate FASTQ files from the Illumina sequencers, is the Tapestry pipeline. This is, again, a web UI that hides a lot of the complexity behind it. This is a tool which will help you upload all of your data to a cloud portal, and on the cloud be able to fire off a pipeline that is going to do mapping of that data, aligning, and then ultimately call the cells that are going to be relevant for variant calling. So this is, again, a simple web user interface where you're logging in, you're uploading your data, creating your samples, and then getting a QC report from that data to say, did the run meet the requirements that I have in terms of cell throughput and the quality of the data before you go into your interpretation mode. This is again available to you for free of charge for all of our customers. We have lots of this data available on our Tapestry Portal, so you can take a look at it today. This tool, I'm not going to spend too much time on other than to kind of go through the back end of it in a little bit, because there's not a lot of interaction here. It's really just a front end to be able to fire off a pipeline, but we wanted to put that onto a web interface so that you didn't have to go worry about getting your own compute infrastructure, getting your own scientists to be able to fire off runs. This is just available to you ready to go when you first get your runs off the Tapestry system. The third tool that we have available is the desktop tool called Tapestry Insights, which allows you to take the variant calling files, load multiple samples together. So this is a multi-sample tool now where if you wanted to do serial time points or some sort of longitudinal analysis, you'd be able to load multiple samples onto this tool. Allows you to review all of the variants that were called, annotate them, filter them based on whether there's prior domain knowledge on the annotations that are interesting or on the specific variant qualities that are high or low quality depending on the thresholds that you want to set, and then ultimately review those variants in the context of how they segregate into subclones. What are the set of cells that have certain mutations, and how are those cells either increasing, decreasing in population, so that you can look at the context of the cell along with the mutations that are relevant or pathogenically interesting. So the Tapestry pipeline tool and the Tapestry Insights tool is a lot of where the bioinformatics analysis is taking place. Similar to what we did in Designer, a lot of the complexity is hidden behind these tools so that you don't have to deal with firing off runs and dealing with the pipelines, and you can really focus more on the analysis and the interpretation that's interesting to you biologically. So what's happening under the hood? In the Tapestry pipeline, this is where you start with a FASTQ file output from the sequencer. First thing we do is we separate all the reads so that we can get the single cell resolution. We identify background from real cells. So this is where we get to our 5,000 to 10,000 cells per run. And then for each one of those cells, we do variant calling. So we're essentially running a variant caller 10,000 times in parallel to be able to call variants per cell. And you'll identify hundreds or thousands of variants based on the type of panel that you're running. Ultimately, that matrix of cells by variants is what goes into the Tapestry Insights tool, where you're going to do the interpretation and analysis over time or over different tissue types. So all of this pipeline is open source tools. We've been using standard tools like BWA and GATK. All of our analysis behind the Tapestry Insights tool is also open source. So we're happy to share this with you and the community. And because really what we want to do is generate data and try to tackle some of the hard problems out there together with everyone in the community. This is not a problem that we're going to solve ourselves. This is not something that we want to keep closed and behind our own walls. We want to share this. We want to collaborate with you and hopefully develop better methods that everybody can use through these simple tools to be able to gain insights into single-cell biology. So just to give you a little bit of a flavor of the user experience, there's a video here that we put together because we can't do a live demo. This is the Tapestry Insights or Designer tool, where you're looking at specific panels, looking at the details of the panels, and then being able to order a panel if you're interested. Here you can bring your own sets of targets to make your own panel if you're interested as well. The second part, this is fairly trivial. You're uploading your FASTQ files, creating your samples, and then selecting the samples that you want to process in the pipeline. This is where the mapping, cell calling, and then ultimately variant calling is being done. The typical run off of a HiSeq is going to take a few hours to run, so you kick it off and you wait for an email to come back to see your results page. So here you can download all of the data, look at a few key metrics that are important to qualify the run before you go into analysis. And the Tapestry Insights tool, again, a desktop tool where you start, load, in this case, multiple data sets. So this is, I think, a time series of 3 time points. So loading the individual 3 files, renaming them so that they're biologically meaningful rather than the random file names that were originally given. You can look at very deep details about the quality of each one of those runs and add some filters with that histogram that you saw. And then here, we've got about 30 different annotation sources that you can look at and filter on to identify the specific mutations that are relevant for your study. Ultimately, those mutations would go into these subclones. And so here, we're naming each of the subclones now something more meaningful because you know which mutations are in each one of those subclones. And then now you can start visualizing these subclones in various ways. Here's an example of how we can look at individual variants across those subclones. This is really interesting if you want to do QC of one variant at a time, or look for examples where you might have loss of heterozygosity or quality. So you can look in detail across subclones, across samples for one variant at a time. Ultimately, this is the thing that most people are going to be interested in, is looking at those subclones over time or over those samples and how they change in relative numbers or absolute numbers. Since we're counting, we can do both. All of this data is then exportable. All of the tables, all of the raw data, all of the figures are exportable so that you can use them for various other tools. So this is just a flavor of how our tool, kind of the user experience behind those tools. Again, all of this, rather than a video, it's much easier to just go look at it yourself. That the tools are available on our website. We also have some example data sets so you can play around with this yourself. There's actually a handful of additional videos and tutorials that we've just started putting up, so there's plenty of material for you to go and check out. So that's a brief look at what our software can do and some of the details behind it. If there's specific questions, I'm happy to take those at the end about how our software works. The last section that I want to cover off on is just to look a little bit into the future about some of the additional things that we're working on. So when we look at our microfluidics device, there are 2 key aspects of it that we really think about that make it powerful for a scalable platform. The scalability allows us to get to millions of drops within minutes. So we can really tag lots of different cells. We have up to, I think now, 2-3 million unique tags that we can tag cells by. So we can scale in the number of cells really fast. The Tapestry Designer tool that you saw also allows us to scale really fast in the number of targets that we can multiplex. We can do tens to hundreds to thousands fairly quickly now, given that we have an automated pipeline that's learning regularly from the data that we're generating. We also think a lot about how this microfluidics device and the ability to put cells into droplets allows us really unique resolution. Because of the way we've designed our microfluidics, the first problem that we tackled was to go after single base resolution, being able to detect SNPs and indels from DNA across thousands of cells. Pair that with the fact that we can do thousands of cells, we can start looking at 0.1% cell population. So we can start looking at 1 in 1,000 cells that might be those rare cells that ultimately give rise to the relapse. The other really interesting thing about the way we've designed our microfluidics is that we retain all of the analytes in a single droplet as it goes through our workflow. So today we're choosing to tag the DNA molecules, but we have RNA and protein as a part of this droplet that goes through the workflow, and we can tag those as well. So I wanted to give you a little bit of a flavor for how this technology scales as we go into the future. And I think what's really exciting for all of us scientists is that this resolution revolution that we're going through with single-cell genomics allows us to access DNA, RNA, protein, being able to really do transomics or multi-omics across these layers to truly gain insights from a systems biology perspective. And that's something that we get really excited about, and we think a lot about how we can do. And in the next few slides, what I want to do is just share with you some of the early data, kind of feasibility data that we have on being able to do multiple analytes from the same cell. Now, these are not products that we have commercially available today, but just to give you a flavor of what we're working on. So this is an example of us being able to look at DNA and protein from the same cell. In this case, we mixed 50-50 two different cell types, Raji and K562. We had two different antibodies specific to each one of those cell lines that we used, along with the AML panel that we've commercialized that have a set of genotypes that are specific to each one of these two cell lines. So in the figure that you see, the scatter plot that you're looking at, each one of the dots is a cell. The x-axis shows you expression of CD19, which is specific to Raji. And then the y-axis shows you expression of CD30, which is specific to K562. And great, we can see a lot of cells line up on either of the axes. That's where the expression is happening. The color coding here, though, is done using the genotypes. There's about 10 mutations in this AML panel that distinguish Raji from K562, and we're using those 10 mutations to color code whether it's a Raji cell or a K562 cell. So again, great validation that CD19 positive cells are also genotyping as Raji cells. So it's a double confirmation that those are really Raji cells versus K562 cells. And what's interesting is when you look at cells that come off of the axes, these are color-coded as blue. These are cells that are expressing both a Raji as well as a K562 protein and have genotypes from both of those cell types. So again, you're getting a double confirmation that these are likely the cell doublets, where both the cells were tagged with the same barcode, and therefore they're having expression of both protein markers as well as genotypes from both cell types. So this is just early feasibility from us showing that we can tag a cell and quantify both the protein expression as well as the genotype from the same cell. Here's some early work where we tried to get both DNA and RNA out of the same cell. In this case, we've got DNA reads coming on the x-axis, and then a specific BCR-able fusion transcript from the RNA coming on the y-axis. So each dot here is, again, a cell, and you can see that you've got a lot of cells that have expression of both the BCR-able fusion transcript as well as reads that are coming from DNA that allow us to do SNP calling or variant calling at the DNA level from the same cell. Again, very early work from us, but just wanted to give you a little bit of a flavor of things that we think about, especially in the context of software that we want to develop on going into the future. So just tying it back to software, we definitely have a lot of volume in terms of the amount of cells that we can generate and the amount of data that our customers are already generating. And this is happening quite fast. It's happening by a variety of different ways, both different panels across different cancer types, as well as now looking at multi-omics, so we're getting DNA, RNA, and protein. And there's still a lot of uncertainty. So you put these four things together, and this makes for a really nice a big data problem where machine learning and AI are an ideal tool to be able to gain insights. So we think a lot about how we can take this data and develop better and better tools using machine learning and AI to make insights into single-cell biology readily available to everyone. One of the last slides that I've got is really more of an advertisement for hiring, collaborating, partnering, call it what you will. This is not a problem we're going to solve all on our own. We think about ways of improving the quality of the data using this big data and machine learning. So things like removing the doublets, dealing with missing data, improving the quality of variant calling and error correcting those variant calls. All of that is to improve the quality so that you better believe the results that you're getting and can make decisions off of that data. And having access to all of the raw data makes it that much better to throw some of the AI methods that are out there towards solving these problems. The last one around multi-omics, we're just scratching the surface here. We have lots of ideas and lots of different paths that we want to explore. And again, we're not going to do it ourselves. So definitely looking forward to hearing from you as to what we could do with this, where should we go with this, how should we develop the tools that give us the insights that we need around multi-omics. So with that, I want to kind of point you all to the Tapestry Portal. You can gain access to all of our software and data sets there. We have documentation there as well, so you can get into more details around what you can do with the software and data interpretation. Very happy to hear from you directly. I've put my email here. So any ideas or ways that we could think about using this data to further research, we're very open to hearing from you on this. So with that, I'd like to thank all of you, thank all of our collaborators for the work. I'll leave with the fact that this is a picture that we took during AML Awareness Day, where we had one AML survivor of 12 years come and talk to us about his personal story going through the disease. One of the questions that he asked us was, what was different about him that allowed him to survive now 12 years past the disease when life expectancy at his age was closer to 5 years, according to most of the statistics? That's a question that we couldn't answer, his doctors couldn't answer, and that's ultimately what we want to be able to do, is really resolve what is the specific difference between patient to patient that allows one to survive and another to relapse. So with that, I will stop and take any questions. With that, if there are any questions, I know there is a question section at the bottom of what you're viewing, so please submit your questions there, and I'm happy to answer them. There's a few there already, but if you don't know how to get to the questions, it's in the bottom list of your browser where you can type in a question. So let me see if I can get through to some of the questions and answer these. All right, so it's a question here going into a little bit more details about how our pipeline works. Yeah, so specifically how our pipeline works, we do use tools like, we have a custom tool for how we call our cells, specifically what's a background versus what's a real cell. But beyond that, we're using a lot of open source tools. So we use FastQC to quality check the files. We use SAM tools for doing some trimming of the reads and such. We use BWA for alignment. GATK is used for our variant calling, and we're exploring many different ways around GATK to improve the quality and specifically optimize it for single cell. So that's an area where there's active development from us. So that's the basics of the pipeline there to get to variant calls. The Insights tool itself is really right now a counting tool where we're just looking at the set of variants, counting across the different subclones to be able to build the fish plots that you looked at. We have in Insights the ability to group those subclones into a phylogenetic tree. We've been collaborating with a couple of groups around looking at different ways of building phylogenetic trees, but it's not something that we've populated into the Insights tool yet. So it's something that is a work in progress. Let's see, maybe moving on to, is there a way to use the pipeline tool command line? Yeah, we're actually in the process of bundling up our whole pipeline that we've deployed on the Tapestry Pipeline cloud solution so that it's available to you as a command line tool to be able to process in your own environment. So another question about how many amplicons can be multiplexed and how big can the panel be. Today on the Tapestry Designer tool, we can multiplex up to 500 amplicons. So this turns out to be about 200 KB of genomic sequence that we can interrogate. We have gone higher than this. We just haven't made it available on the Tapestry Designer tool yet. It's something that we can definitely have a conversation about to go higher if needed. The real challenge ends up becoming how much cost to sequencing is possible. As we make the panels bigger, the cost gets much higher. And let me see if I can see another question. It says, great relevance in cancer analysis, but do you see any projects analysis with other rare circulating cell types, like circulating fetal cells? Yeah, that's a great question. There are definitely other areas that we've thought about. Circulating cells is one that we think about both from circulating cancer cells as well as, in this case, you mentioned fetal cells. We're working on improving the capture of our cells so that we can go after really rare cell types that can be input into our system. So it's definitely an area that we think about and would be great to chat specifically about the application that you're thinking about to see if we can enable that today. We can make a panel for pretty much any set of genes, so that's not the limiting factor now. It's can we get the cell type into the system? And we've got some pretty promising data being able to start with a very few number of cells to be able to interrogate at a single cell level. The other area that we think about in terms of, or outside of cancer analysis, is in the neurological diseases. There's a lot of somatic mutations there, and we think about how those somatic mutations in neuronal cells could be elucidated with a technology like this. All right, let me... questions about dinucleotide and trinucleotide variations as well as CNVs. So dinucleotide and trinucleotide variations, I think we can do that today. That's not a question that I'm absolutely sure I can answer. But also on the copy number, we've definitely seen signal in our data that demonstrates that copy number is a possibility with this amplicon-based method. It's something that we're actively looking at collaborations around to see if we can identify the right system to optimize an algorithm around. So we absolutely think copy number is possible. It's a matter of partnering and developing a copy number algorithm that's specific to single cell. So definitely would like to look into it. There was a question also around future methods around more robust clustering, dimensionality reduction, and looking at many variants at a time rather than the handful of variants that we can select through filtering today. Yeah, we actually have this as an active project today. We're definitely looking at how we can, as the panels that we're studying get bigger and bigger, there are more and more mutations that people want to look at. We're looking at ways that we can do multi-dimensional scaling, clustering across these multiple dimensions, and visualizing data similar to how we've been doing it in single-cell RNA using these dimension reduction algorithms. So it's an active area for research for us today. So definitely you'll be seeing more of that from us. So question around how do you do sample prep for solid tumors? Do you have any automated procedures for this? Sample prep on solid tumors, so our system today can take as input whole cells or nuclei from various different sources like fresh frozen tissue, cryopreserved tissue. We've tested probably 40 or 50 different cell types to date. This is something that we're starting to expose on that Tapestry Portal, so have a look at that to see how many other sample types we can support. For solid tumor, we've seen that the simplest way to do it is to extract nuclei and put nuclei into the system. That eliminates the tissue type to tissue type variability and having to optimize the sample prep for each tissue type. Have we automated this? We have yet to look at ways to automate it. To date, most people have been doing this manually. What sequencers are compatible with the Tapestry platform? We pretty much support all of the sequencers. We had a big effort at the end of last year to make the pipeline support all of the different sequencers. So everything from MySeq to NextSeq to HiSeq and NovaSeq are supported now. So it shouldn't be an issue in terms of running the system. We know that there's variation from sequencer to sequencer, so that's something that you will have to be aware of, like NextSeq, especially with some of the older chemistries, have higher error rates, so that might not be the best sequencer for rare cell detection. But otherwise, the pipeline supports all of the different sequencers. Question around how long does it take to design custom panels? The custom panel tool, it's a matter of minutes. Maybe it takes an hour or so to design those custom panels, so it's actually fairly fast turnaround time to develop a custom panel. We've baked in the entire human exome into the back end of the tool. So if you're trying to design something from one of the genes in the human exome, it should be a matter of minutes to get you a panel design that you can evaluate. Let's see, any other questions? There was a question here around how do we track the zygosity of mutations? So given that we're doing everything at the single cell level, each mutation is called independently in every cell. We can start calling, essentially germline variant calling in each of the cells, and therefore you can look at homozygous versus heterozygous mutations, and then because you're doing it at each cell level, you can then just sum up the number of cells. So zygosity can be tracked at an individual cell level, and then the kind of the somatic variant allele frequency that we're looking at is really just a sum of all of the cells that have a specific homozygous or heterozygous mutation. So it makes it really powerful for, especially in the gene editing field, being able to see if the gene editing system was targeting edits to both alleles or just one allele. All right, I think answered most of the questions here. So with that, I thank everyone for attending this, and please contact us if there are any other ideas, questions, or discussions that you want to have with us. Thank you very much for your time, and we'll talk to you again, I think, in another couple of months with another webinar, I think. Great, thanks.